Hi everyone, this is Professor Beda Ahmed, and uh, chapter 20, the heart. The heart, along with the blood and blood vessels, is a part of the cardiovascular system. This system function to distribute oxygen and nutrient to the cells of the body and take away carbon dioxide and other wastes. In this chapter, we consider the structure and function of the heart, a small organ about the size of a person's clenched fist. And this extraordinary organ beats approximately 100,000 times each day. Each day, the heart pumps about 2,000 gallons, or just over 9,000 liters of blood. So the heart is a part of the cardiovascular system, which consists of the heart, blood, and blood vessels. And the type of blood vessels are arteries. Arteries carry blood away from the heart. Veins, which return blood to the heart. Capillaries are called exchange vessels. Microscopic thin walled, the capillaries, and these are microscopic thin walled vessels interconnect the smallest arteries and the smallest veins, exchange nutrients and dissolve gases called gas exchange, and waste between the blood and surrounding tissues occurs through their thin walls. So the cardiovascular system consists of heart, blood vessels, and in the cardiovascular system, blood flows through a network of blood vessels that extend between the heart and peripheral tissues. Those blood vessels make up a pulmonary circuit, which carries the blood to and from the gas exchange surfaces of the lung, and consists also of systemic circuits which transport blood to and from the rest of the body. Each circuit begins and ends at the heart, and the blood travels through these circuits in sequences. Uh, let's see heart location and position. The heart is in the thoracic cavity near the anterior chest wall, directly posterior to the sternum. The great vessels, both veins and arteries, are connected to the superior end of the heart at its base, and the inferior pointed tip of the heart is apex. It's called apex. The heart sits in the interior portion of the mediastinum cavity, the region between the two pleural cavities. Uh, outer outer covering of the heart is called the pericardium. Pericardium surround the heart and consist of an outer fibrous uh, pericardium and an inner serous pericardium. The inner serous pericardium is a two-layered membrane composed of outer parietal layer and an inner visceral layer. And the visceral layer, also called epicardium, the potential fluid-filled space between these two serous layers in the pericardial cavity, which contain 15 to 50 ml of pericardial fluid secreted by pericardial membrane. And these fluids act as a lubricant, reducing friction between the opposing visceral and parietal surfaces as the heart beats. Pathogens can infect the pericardium, producing inflammation um, and this condition called pericarditis. Um, the inflamed pericardial surface rub against one another, making a distinctive scratching sounds called a friction rub that can be heard through an instrument called a stethoscope that is placed on the chest. The pericardial inflammation also commonly results in increased production of pericardial fluid. Fluid then collects in the pericardial cavity, restricting the movement of the heart. This condition called cardiac tamponade can also result from traumatic injuries such as stab wounds that produce bleeding into the pericardial cavity. Superficial anatomy of the heart. The two atria have relatively thin muscular walls and are highly expandable. And when not filled with blood, the outer portion of each atrium deflats and becomes a lumpy, wrinkled flap. This expandable extension of an atrium is called an auricle. The sulci, deep grooves, um, which the coronary sulcus marks the border between the atria and the ventricles. The anterior interventricular sulcus and the posterior interventricular sulcus are shallow depressions that mark the boundary between the left and right ventricles. The three layers of the heart wall, a section through the wall of the heart reveals three distinct layers. The visceral layer of serous pericardium or epicardium covers the surface of the heart. This serous membrane consists of an exposed mesothelium and an underlying layer of areolar connective tissue that is attached to the myocardium. The parietal layer of serous pericardium consists of an outer dense fibrous layer, an areolar layer, and an inner mesothelium. And then myocardium, the second layer, myocardium is cardiac muscle tissue that forms the atria and ventricle. This muscular layer contains cardiac muscle cells, connective tissue, blood vessels, and nerves. And then the endocardium covers the inner surface of the heart, including those of heart valves. It is made up of simple squamous epithelium and underlying areolar tissue. There are four chambers of the heart. The right atrium receives blood from the systemic circuit. The right ventricle pumps blood into pulmonary circuit, and the left atrium receive blood from the pulmonary circuit. The left ventricle pump blood into systemic circuit. The right atrium receive blood from the systemic circuit through two great veins, the superior vena cava, opens into the posterior and superior portion of the right atrium at and it delivers blood to the right atrium from the head, neck, upper limbs, and chest. The inferior vena cava opens into the posterior and inferior portion of the right atrium. It carries blood to the right atrium from the rest of the trunk, viscera, and lower limbs. And note that there are no valves between the vena cava and the right atrium. Right atrium, the right atrium from the 5th week of the embryonic development until birth, there is an oval opening called foramen ovale, penetrate the internal or interatrial septum and connects the two atria of the fetal heart. Before birth, the foramen ovale permits blood to flow from the blood or the right atrium to the right or to the left atrium. So again, before birth, the foramen ovale permits blood to flow from the right atrium to the left atrium while the lungs are developing. At birth, the foramen ovale closes and the opening is permanently sealed off within 3 months of delivery. If the foramen ovale doesn't close, serious cardiovascular problem may result. A small shallow depression called the fossa ovalis remains at the site of adult heart. The right ventricles need less pressure than the left ventricle because of the circulation paths they serve. Right ventricle pulmonary circulation pumps blood to the lungs, which are very close to the heart. The pulmonary arteries and lungs have low resistance to blood flow, so only small pressure is needed to push blood through. If the pressure were too high, it could damage the delicate capillaries in the lungs. The left ventricle, in which push blood to the systemic circulation, pumps blood to the entire body. The systemic arteries and tissues have much higher resistance than the lungs. So the left ventricle need to generate a much higher pressure to push blood throughout the body. This is why the left ventricular wall is thicker and more muscular. Both pump the same volume of blood. Right ventricle, thinner walls, a lower pressure, pouch shape pumps blood only to the lungs, which is a short distance, a low resistance, and left ventricle, thicker muscular wall, higher pressure, rounder shape, pumps blood to the whole body, long distance, and high resistance. It needs high resistance. The left ventricle is stronger because it must push blood much farther than the right ventricle. The chambers of the heart separated by muscular partitions called septa or septum for singular. The atria are separated by the interatrial septum, and the ventricles are separated by the much thicker interventricular septum. The heart also has valves, covered opening that direct the flow of blood between chambers and vessels. Sorry. The cardiac skeleton stabilizes the positions of these valves, and the two atrioventricular valves called tricuspid and mitral bicuspid are folds of fibrous tissue that extend into the opening between the atria and ventricles. These valves permit blood to flow only in one direction from the atria to the ventricles. There are also two semilunar valves, pulmonary and aortic vessels, um, which between the ventricles and their great vessels, which ensure blood flows into these vessels. Blood travels from the right atrium into the right ventricle through a broad opens, bordered by the three fibrous flaps. These flaps, called cusps, are part of the tricuspid valve, also known as the right atrioventricular valve. The free edge of each cusp is attached to connective tissue fibers called chorditendinae, and with the chorditendinae fibers originate at the papillary muscles, conical muscular projections that arise from the inner surface of the right ventricle. The atrioventricular valves prevent the backflow of blood from the ventricles to the atria when the ventricles are contracting. When the ventricles contract, blood moving back toward the atria swings the cusps together, closing the valves. At the same time, the contraction of the papillary muscles tenses the chorditendinae, stopping the cusps before they swing into the atria. So if the chorditendinae were cut or the papillary muscles were damaged, backflow called regurgitation of blood into the atria would occur each time the ventricle contracted. The right ventricle, we have the internal surface of the right ventricle contains series of muscular ridges. These muscular ridges called trabeculae carneae. And trabeculae carneae, those are internal surface of the right ventricle contain, as I said, series of muscular ridges. And the moderator band is a muscular ridge that delivers the stimulus from contraction to the papillary muscles. As a result, they begin tensing the chorditendinae before the rest of ventricle contracts. The coronary circulation. Coronary circulation supplies blood to the muscle tissue of the heart. The left and right coronary arteries originate at the base of the ascending aorta, at the aortic sinuses. Blood pressure here is the highest in the systemic circuit when the left ventricle contracts and forces blood into the aorta. The high pressure of this blood stretches the elastic walls of the aorta. When the left ventricle relaxes, blood no longer flows into the aorta. Pressure decline and wall of aorta recoil. This recoil, called elastic rebound, pushes blood both forward into the systemic circuit and backward through the left and right aortic sinuses, and then into the respective coronary arteries. In this way, the combination of elevated blood pressure and elastic rebound ensure a continuous flow of blood to meet the demand of active cardiac muscle tissue. The right coronary artery flows the coronary sulcus, and around the heart. It supplies blood to the right atrium, then a portion of both ventricles, and portions of electrical conducting system of the heart. The right coronary artery generally gives rise to one or more marginal arteries and the posterior interventricular artery. Cardiac veins, the great cardiac vein begin on the anterior surface of the ventricles along the interventricular sulcus. This vein drain blood from the region supplied by the anterior interventricular artery, which is a branch of the left coronary artery. The cardiac veins draining the myocardium return blood to the coronary sinus, which is a large, thin-walled veins opens into the right atrium. Other cardiac veins empty into the great cardiac vein or the coronary sinus. These veins include posterior vein of the left ventricle, draining the area served by the circumflex artery, the middle cardiac vein, um, draining the area supplied by the posterior interventricular artery. The small cardiac vein, which receives blood from the posterior surface of the right atrium and ventricle, and the anterior cardiac vein, also called the anterior veins of the right ventricle, which drain the anterior surface of the right ventricle, empty directly into the right atrium. Coronary artery disease is the buildup of plaque uh, causing partial or complete blockage of the coronary circulation. Cardiac muscle cells need a constant supply of oxygen and nutrients, so any reduction in blood flow to the heart muscle produce corresponding reduction in cardiac performance. And you only need to know the first three issues or factors that affect the coronary blood vessels. In a single cardiac contraction called the heartbeat, all heart chambers contract in a series. First, the atria and then the ventricles. Two types of cardiac muscle cells are involved in a normal heartbeat. The first one is a specialized autorhythmic cells, pacemaker and conducting, both, and of the conducting system control and coordinate the heartbeat. And then we have contractile cells. Contractile cells produce the powerful contractions that prepare blood. Each heartbeat begins with an action potential generated by cells of conducting system. This system is a network of specialized cardiac muscle cells that initiate and distribute the stimulus to contract. Unlike skeletal muscle, cardiac muscle tissue contracts on its own without neural or hormonal stimulation. This property is called autorhythmicity. Many clinical problems result from abnormal pacemaker function. Bradycardia is a condition in which the heartbeat is slower than normal, and tachycardia is faster than normal heart rate, and ectopic pacemaker in abnormal conducting cell or ventricular contractile cell begin generating action potential at a higher rate. The activity of an ectopic pacemaker partially or completely bypass the conducting system, disturbing the timing of ventricular contraction. The result may be dangerous reduction in the pumping efficiency of the heart. The network of specialized cardiac cells of the conducting system are grouped in various locations. Pacemaker cells are essential to establishing the normal heartbeat. They are also called nodal cells because they are found in two nodes or locations. The sinoatrial nodes. A sinoatrial node is embedded in the posterior wall of the right atrium near the entrance of the superior vena cava. Because this sino or SA node is the primary driver of the heart rate, it is also known as the cardiac pacemaker. So the relatively large atrioventricular node is located at the junction between the atria and ventricles near the opening of the coronary sinus. The pacemaker cells of this node send signal from the cells of the sinoatrial node and act as a backup to the sinoatrial node pacemaker cells. Conducting cells interconnected the sinoatrial and AV nodes or atrioventricular node and distribute the contractile stimulus through the myocardium. These cells are found in two main locations. In the atria, conducting cells are found in internodal pathways in the atrial walls. These pathways distribute the contractile stimulus to the surrounding atrial muscle cells. As this electrical impulse travels from the sinoatrial node to the AV node, the conducting cells include those in the atrioventricular bundle. In the ventricle, we have bundles, also called bundle of Hess, and the bundle branches, branch that run between the ventricle as well as the Purkinje fibers, which distribute the stimulus to the ventricular myocardium. Now let's trace the path of an impulse from its initiation at the SA node, a sinoatrial node, examining its effect on the surrounding myocardium as we proceed. Functions of pacemaker cells, sinoatrial at first, number one, the sinoatrial node activity and atrial activation begin. A stimulus speeds across atria and reach an AV nodes, atrioventricular node, and then impulse is delayed from a sinoatrial node to AV node, and then to the atrial contraction begin. The atrial contraction begin. This impulse is delayed for 100 milliseconds at the atrioventricular node. Then the impulse travels in atrioventricular bundle to the left and right, both bundles, and then branches in interventricular septum to Purkinje fibers and papillary muscle via moderator band. And then the impulse is distributed by Purkinje fibers and relayed throughout the ventricular myocardium. Atrial contraction is completed and ventricular contraction begin. The electrical events in the heart are powerful enough to be detected by electrodes on the body. A procedure known as electrocardiography can monitor the electrical events of the conducting system. A recording of these events is electrocardiogram, also called ECG or EKG. Clinicians can use an ECG to assess the function of a specific pacemaker, conducting, and contractile cells. When a portion of the heart has been damaged by heart attack, for example, the ECG reveals an abnormal pattern of impulse conducting, and the monitoring electrodes for an ECG are placed at different locations on the body surface. The small p wave, these are the features of ECG. The small p wave, which accompanies the depolarization of the atrial contractile cells, and the QRS complex appears as ventricle contractile and depolarization. This electrical signal is relatively strong because the ventricular muscle is much more massive than of the atria. The ventricles begin contracting shortly after the peak of the R wave. The smaller T wave, which indicate repolarization of the ventricular contractile cells, the times between waves are reported as segments and intervals. Segments generally extend from the end of one wave to the start of another wave. Commonly used segments and intervals are the PR interval, extend from the start of atrial depolarization to the start of QRS, and which is the ventricular depolarization. And the QT interval indicate the time required for the ventricles to undergo a single cycle of depolarization and repolarization. The Purkinje fibers distribute the stimulus to the cardiac contractile cells, um, which form the bulk of the atrial and ventricular walls. These cells account for about 99% of the muscle cells in the heart. Cardiac contractile cells are interconnected by intercalated disc. The resting membrane potential of a ventricular contractile cell is approximately 90 millivolt, comparable to that of the resting skeletal muscle fiber, which is minus 85 millivolt. The resting membrane potential of an atrial contractile cell is about minus 80 millivolt. Cardiac contractile cells are interconnected by intercalated disc, okay? And the intercalated disc transfer the force of contraction from cell to cell and propagate action potential. At an intercalated disc, the interlocking membrane of adjacent cells are held together by desmosomes and linked by gap junction. Cardiac contractile cells, they are small in size. They are single centrally located. They have single centrally located nucleus and branching interconnected between cells. They are branched, and they also the hallmark of cardiac muscle cells, the presence of intercalated disc that play a role in action potential conducting. The process of action potential in contractile cells rapid depolarization. A massive influx of sodium ions rapidly depolarizes the sarcolemma through fast sodium channels, and the plateau extracellular calcium ions enters cytosols through slow calcium channels, and repolarization. Potassium ions rushes out of cells through slow potassium channels. The refracting period, the membrane of the cardiac contractile cells will not respond normally to a second stimulus for some time, and after an action potential begins, this time is called the refractory period. The ventricular contractile cells cannot respond. The absolute refracting period lasts approximately 200,000 or 200 milliseconds. And this absolute refracting period is followed by a shorter 50 millisecond relative refractory period. The membrane will respond to a stronger than normal stimulus by initiating another action potential. In total, an action potential in a ventricular contractile cell lasts 250 to 300 milliseconds, about 30 times as longer as a typical action potential in a skeletal muscle fiber. The appearance of an action potential in the cardiac contractile cell plasma membrane produce a contraction by causing an increase in the concentration of the calcium ion around the myofibrils. This process takes place in two steps. Extracellular calcium ion crossing the plasma membrane during the plateau phase of the action potential provide roughly 20% of calcium required for a contraction. The arrival of extracellular calcium ion triggers the release of additional calcium ion from reserve in the sarcoplasmic reticulum. Extracellular calcium ions thus have both direct and indirect effects on cardiac contractile cell contraction. For this reason, cardiac muscle tissue is highly sensitive to changes in the calcium ion concentration of the extracellular fluid. As the slow calcium channels close, the intracellular calcium ion are pumped back to the sarcoplasmic reticulum or out of the cell, and the contractile cell relaxes. While a normal heart is beating, it gets energy as the mitochondria break down fatty acids, which is stored, fatty acids stored as lipid droplets, and glucose is stored as glycogen. These aerobic reaction can occur only when oxygen is readily available. In addition to obtaining oxygen from the coronary circulation, cardiac contractile cells maintain their own sizable reserve of oxygen, and in these cells, oxygen molecules are bound to the heme units of the myoglobin molecules. A brief resting phase follows each heartbeat, allowing time for the chambers to relax and prepare for the next heartbeat. The period between the start of one heartbeat and the beginning of the next is a single cardiac cycle. It's called cardiac cycle, and it includes alternating periods of contraction and relaxation. For any one chamber in the heart, the cardiac cycle can be divided into two phases, systole and diastole. During systole, a contraction, this is the contraction. The chamber contracts and pushes blood into adjacent chamber or into an atrial trunk. And systole is followed by diastole or relaxation. During diastole, the chamber fills with blood and prepares for the next cardiac cycle. Fluids flow from an area of higher pressure to an area of lower pressure. And in cardiac cycle, the blood pressure within each chamber rises during the systole and falls during diastole. The correct pressure relationship depends on the careful timing of contractions and valves between um, the valves between adjacent chambers help ensure that blood flows in the required direction. The phases of the cardiac cycle: arterial systole, arterial diastole, ventricular systole, and ventricular diastole. For a representative heart rate of 75, uh, at this heart rate, a sequence of systole and diastole in either the atria and the ventricular lasts 800 milliseconds. When the heart rate increases, all the phases of the cardiac cycle are shortened. The greatest reduction occurs in the length of time spent in diastole. Listening to the heart, a technique called the listening to the heart, this technique called auscultation, is a simple and effective method of cardiac assessment. And there are four heart sounds named S1 through S4. Listening to the heart with a stethoscope can detect the first and second heart sound. These sounds accompany the closing of the heart valves. The first heart sound, S1, known as loop, lasts a little longer than the second, S2, called do. And S1 marks the start of ventricular contraction when the AV valves close and semilunar valves open. S2 occurs at the beginning of the ventricular filling when the semilunar valves close and the atrioventricular valves open. The third and fourth heart sounds are usually very faint and are seldom audible in healthy adults. These sounds are associated with the blood flowing into the ventricles, S3, and atrial contraction, S4. Rather than the valve action, if the valve cusps are malformed or there are problems with the papillary muscles or chordae tendineae, the heart valves may not close properly. And then atrioventricular valve regurgitation then occur, and this occur during a ventricular systole. If the valve cusp or malformed, or there are problems with the papillary muscles or chordae tendineae, the heart valves may not close properly, and an AV valve regurgitation then occurs during ventricular systole. The surges, swirls, and eddies of turbulent blood flow that accompany regurgitation create a rushing, gurgling sounds known as a heart murmur. Um, when considering cardiac functions over time, physicians generally are most interested in the cardiac output, the amount of blood pumped by the left ventricle in 1 minute. Um, in a sense, cardiac output is an indication of blood flow through peripheral tissues, and without adequate blood flow, homeostasis cannot be maintained. The cardiac output provides a useful indication of ventricular efficiency over time. We can calculate cardiac output by multiplying the heart rate by the average stroke volume, which is CO, cardiac output, and heart rate, HR, and then stroke volume. Let's look into stroke volume. Uh, volume, as it is the most important factor in examination of a single cardiac cycle. Stroke volume can be expressed as SV equal to EDV, which is ESV subtracted from EDV. EDV is end diastolic volume, and ESV is end systolic volume, and EDV is the amount of blood in each ventricle at the end of ventricular diastole, and ESV is the amount of blood remaining in each ventricle at the end of ventricular systole. And the ejection fraction is the percentage of EDV that is ejected during ventricular contraction. Under normal circumstances, autonomic activity and circulating hormone make homeostatic adjustment to the heart rate as cardiovascular demands changes, thereby adjusting cardiac output. These factors act by modifying the autorhythmicity of the heart. Even a heart removed from a heart transplant continues to beat unless it is kept chilled in preservative solution. Many hormones affect heart contractility, like epinephrine, norepinephrine, thyroid hormone, um, and other hormones. Many hormones affect heart contractility. Um, Many of the drugs used to treat hypertension, high blood pressure are negatively inotropic, stimulate or block alpha and beta receptors, or block calcium ions channels. The purpose of cardiovascular regulation is to maintain adequate blood flow to all body tissues. The heart cannot do this by itself, and it doesn't work in isolation. So, the cardiac centers adjust not only the heart rate, but also the diameters of peripheral blood vessels. These adjustments work together to keep the blood pressure within normal limits and to maintain circulation to vital tissues and organs. The cardiovascular responses to changing activities and circulatory emergency.